October 25th, I believe, right? Or is today 25th? Yeah, 23rd, October 23rd. The second part of the running case study is available. Please have it printed off so you can turn it in Monday. Don't email it to me. Bring in a printed sheet. If you don't trust the printers here, make sure you print it off before you come to class. So you should probably do it, I would say, Friday or Saturday anyway, because it's going to be material that's on the exam. It deals with coagulation and clotting. So print that off and turn it in on Monday on your way in. Give it to one of the TAs. There are 2 practice exams. One is all cardio questions, and one is almost all respiratory questions, except there's a Wigger's diagram that I stuck in at the end of it. So I will give the TAs answer keys for those. So if you have questions you can't figure out the answer to, go to one of the review sessions, either Thursday or Sunday, and get clarification. Or if you can't make the review session, send me an email. Don't just send me an email that says, what's the answer to question 15? Because then I'll send you back an email that says, I don't know, man. You tell me. Right? If you're going to ask me the answer to a question, make it seem like you've actually struggled with the question in your mind and say, well, I think it's this answer because, and then I'll tell you if you're right or wrong, and I'll tell you the right answer. Okay? I want to make sure that you're actually going through and doing these and running it through your mind. It'll help you when it comes exam time. Any questions on anything? All right. So we've been talking about the respiratory system. We're going to talk about how it is regulated. So regulation of respiration. Respiratory control centers in the medulla, which is a common place for control centers. It's where we have the cardio-inhibitory center, the vasomotor control center that we talked about. There are some areas in the pons that we will talk about, pneumotaxic area. Basically, respiratory drive, battery's running low, is going to come from this respiratory control center in the medulla. So that's going to give you your rhythmic breathing. Normal breathing rate is usually between 8 to 18 breaths per minute. And it can increase up to 40, 50 breaths per minute during heavy exercise. Now, this respiratory control center can be influenced by a number of different things. You can voluntarily choose to hold your breath if you're mad at somebody, like your parents. I'm going to hold my breath. Or you can voluntarily hyperventilate if you're making an obscene phone call. Emotional stimuli. Some people are prone to hyperventilation if they're overly stimulated emotionally. Anxiety in people with asthma can sometimes trigger an asthma attack. Chemoreceptors, peripheral and central. Peripheral chemoreceptors are located the same place where we find baroreceptors, in the aortic arch and the carotid bodies. The peripheral chemoreceptors are most sensitive to oxygen. If I were to order what's up there on the screen, I would have changed it so that it goes in the order O2, pH, CO2. Yay! because the peripheral chemoreceptors are most sensitive to oxygen. Central chemoreceptors are most sensitive to carbon dioxide. Central chemoreceptors are located in the medulla, right next to the neurons that are controlling respiration. You have other reflexes, like the Herring-Brewer reflex. You have stretch receptors in your lungs so that you do not overinflate your lungs. As you stretch your lungs, there's an inhibitory signal that goes back to the medulla to decrease inspiration. Proprioceptors in the muscles. So when you when you increase activity, say during exercise, you will increase your breathing even before blood CO2 and O2 change because of that increase in proprioceptor activity. It's kind of an anticipatory event. You're exercising, breathe more. And then certain stimuli, temperature, pain, touch. Let's try this again. Hey, there we go. So when we deal with breathing, we have a period of inspiration where you're breathing in, expiration where you're breathing out. That cycle, again, is influenced by higher centers, voluntary control, and reflexes, right? Chemoreceptor reflexes, stretch receptor reflexes, etc. And the whole idea here, controlling the cycles of inspiration and expiration, is going to control the contraction and relaxation of the muscles associated with breathing. Right, so the diaphragm, which is controlled by the phrenic nerve, and then the other muscles, right, the internal and external intercostals, the abdominal muscles, those muscles of breathing. So we're going to deal with sensors, central controller, and effectors. So we're basically talking about reflexes. Respiratory center here. Dorsal and ventral respiratory groups. Dorsal group is primarily involved in breathing in. Primarily contains neurons that are called I neurons, I for inspiration. Ventral respiratory group contains both I and E neurons. So as you can probably guess, most of the control for respiration revolves around inspiration. Revolves around contracting the diaphragm and relaxing the diaphragm. A couple of other centers. We're not going to talk about the apneustic center. Don't know much about it. We do know that when it's stimulated, it causes you to take a deep breath, stop breathing for a period of time, and then take another breath. So kind of apnea, apneic breathing. Pneumotaxic center, which seems to be involved in regulating the cycles of inspiration and expiration. What we basically have here, if we look at neuronal discharge from the dorsal root, I wanted to say dorsal root ganglia. Aha! Dorsal respiratory group. Different DRG. So as these neurons fire action potentials, the nerve activity in the phrenic nerve, the nerve that controls the diaphragm, is going to increase to cause contraction and then decrease, which will cause relaxation. So this gives you this kind of burst pattern here in the phrenic nerve. So the pacemaker of the heart is what? The SA node. Well, the pacemaker for breathing is called the pre-Botzinger complex, or the pre-Bots. The neurons within the pre-Botzinger complex are similar to the cells in the SA node in that they do not have a stable resting membrane potential. The thing is, they don't depolarize as quickly as the SA nodal cells. They depolarize more slowly, hence your breathing rate of about 10 breaths per minute rather than a heart rate of 70 or 80 beats per minute. But it's the same principle. Unstable resting membrane potential causing a burst of neuronal activity, repolarization, etc. Now this rhythm is going to be altered by the reflexes we're going to talk about. Spend most of our time with the chemoreceptor reflex because that's the most important one. Also tends to be the most difficult. Stretch receptors, joint receptors we'll discuss a bit. Other respiratory centers, well, we already talked about all of them. Lung receptors, different types of lung receptors. Stretch receptors, that's part of the Herring-Brewer reflex, so that you don't overstretch and damage the tissue. Irritant receptors, we're all familiar with irritant receptors. They're found throughout the respiratory tract. Bronchioles, trachea, back of the throat. So stimulating these irritant receptors causes you to cough or to go clear your throat, whatever. Something is stimulating the irritant receptors, dust, mucus, whatever, causing you to want to clear that blockage and reestablish the tube. Could lead to bronchoconstriction if they are stimulated enough. And the idea behind that would be to prevent anything that's in the conducting passages getting down into the alveoli and interfering with gas exchange, so producing more dead space. So you cough or you sneeze in order to get rid of or expel the irritating substance. J receptors, juxtacapillary receptors, they're located in the capillary walls, the capillaries that are traveling through the lung tissue. They're sensitive to stretch. So that's where your part of your Herring-Brewer reflex comes in. Other things, chemoreceptors, we're going to spend a fair amount of time talking about. Baroreceptors, elevated blood pressure, can stimulate brief periods of apnea, bronchodilation. And then muscle and tendon reflexes in order to adjust respiration based upon needs or elevated workloads. I'm having a problem with batteries today. All right. Moving on. So these are the fibers in the vagus nerve that are associated with the various receptors. All right, so slowly adapting receptors, found primarily in airway smooth muscle. They're responsible for sensing inflation, for the Herring-Brewer reflex. That's going to cause a shortening of inspiratory time. So as you inhale, as you inflate your lungs more, there's going to be less and less of an inspiratory signal coming from the ventral respiratory and the dorsal respiratory groups. Rapidly adapting receptors. Those are sensitive to a variety of different irritating substances. Unmyelinated C fibers, so small C fibers. So these fibers here were more likely in the A delta region. Unmyelinated C fibers. These are the fibers that are going to be sensitive to capsaicin. If you've ever cooked with chili peppers or something and you heat the pan first and you throw the chili peppers in and then you breathe in, you have a brief period of apnea where you can't breathe because the capsaicin that you just breathed in has just completely saturated those receptors. So when you do that, throw the chilies in before you heat up the pan, or if you have to do it when the pan's heated up, have somebody else do it, or stand away, or have a good ventilation system, something. Generation of respiratory rhythm lies where? Where do we get our respiratory rhythm from? Yes? Yeah, if you inhale anything that contains capsaicin, it can cause you to have a brief apneic period, a brief period where you can't breathe. All right, hold them up. Let me see what you got. All right, most of you got D, Cree-Botzinger complex, the Cree-Bots. So now we're going to talk about chemical control of respiration. By far the most important chemical regulator of respiration is CO2. If you recall, if you looked at the Camtasia that I recorded for Monday, there's such a big reservoir of oxygen bound to hemoglobin inside of erythrocytes that you can withstand 20, 30, 40% decreases in oxygen content in the blood and still be fine. But small changes in CO2 can be devastating. CO2 is a volatile acid. It is converted to carbonic acid, H2CO3. So not only does it affect the ability of tissue cells to get rid of oxygen, but it also alters the pH of body fluids. And you alter the pH of body fluids, you alter the three-dimensional structure of proteins, which alters their ability to function properly, particularly enzymes. And if you deform them enough, they do not function at all, and you die. And that is bad. I've heard, but I've never tried it. We'll talk about mostly carbon dioxide, a little bit about hydrogen ions, and some about oxygen. So we've got central and peripheral chemoreceptors. So the central chemoreceptors located in the medulla next to the neurons that control respiration. Primarily sensitive to changes in CO2. I don't like this diagram. I'll show you why I don't like this diagram in a little bit. And then peripheral chemoreceptors, primarily sensing oxygen. Aortic body, located where the baroreceptors are, and in that notch, the bifurcation of the external and internal carotid arteries, carotid bodies. Again, same place, you'll find peripheral baroreceptors. Here's why I did not like that diagram. What happens is you have a blood-brain barrier, which we talked about in neuro, and you've probably talked about in PDA. Hydrogen ions cannot cross that blood-brain barrier. They are charged molecules. Charged molecules don't cross the barrier very well. In order to cross that barrier, you have to be lipid soluble or very weakly charged. So hydrogen ions in the blood aren't going to affect the central chemoreceptors. The only thing that can happen is CO2, which is lipid soluble, can cross the blood-brain barrier, and then in the cerebrospinal fluid, in the interstitial fluid surrounding the neurons, you convert CO2 into carbonic acid, H2CO3. That disassociates into hydrogen ion and bicarbonate. So what is actually being sensed by the sensors in the central chemoreceptive area is a change in interstitial fluid pH in the brain. So it's directly sensing hydrogen ions in the interstitial fluid. But that's an indirect measure of how much CO2 is in the blood. Right? Because the more CO2 is in the blood, the lower the pH is going to go. The less CO2 in the blood, the higher the pH is going to go. Now, cerebrospinal fluid is a low-protein environment. There's not a lot of protein in CSF. The only time you'll find protein there usually is if you have an infection like meningitis or some type of damage to the meninges. pH is normally a little lower than blood plasma, and CO2 content's normally a little higher than blood plasma, and that's because the brain is always working. It's always metabolically active. You don't have periods where your brain is not metabolically active like you do for other tissues. The brain's always working. When you're sleeping, it's still working. It's just that different areas are working during sleep that don't work so hard when you're awake. But brain metabolism is pretty much constant. Peripheral chemoreceptors, found in the carotid bodies, respond most greatly to oxygen and a little bit to carbon dioxide and a little bit to hydrogen ion. But if carbon dioxide changes, the central chemoreceptors are going to sense that before the peripheral chemoreceptors are able to respond. As you can see, their greatest response rate is found here at a PO2 of below 100. Right? So normal arterial PO2 is 100. As it drops, you start to get an increase in discharge frequency from those sensors, which then gets carried up the glossopharyngeal or vagus nerves to the respiratory control center. These sensors, oxygen sensitive, so if you get a decline in PO2, you'll get a decline in production of ATP, which will close ATP-sensitive potassium channels. What happens when you close potassium channels? Bueller. I know a good test question. 50-50 shot. D, depolarization. Right? Potassium cannot leak out of the cell. It accumulates. It stays there. Slowly increases towards threshold. Threshold in this case, calcium channels, voltage-gated calcium channels. When calcium comes in, triggers exocytosis of ATP and acetylcholine. That's going to stimulate the afferent fibers, the neurons, that are going to take information either via the glossopharyngeal nerve for the carotid bodies or the vagus nerve for the aortic arch, up to the respiratory control center in order to alter breathing, inspiration and expiration. So here we see these glomus cells, which are the sensory cells that have the oxygen sensors on them. And then this happens to be cells that are in the carotid bodies because they're sending or they're stimulating afferent neurons that are in the glossopharyngeal nerve. This was the carotid, or excuse me, this is the carotid. If this were the aortic, it would be the vagus nerve. Questions? You're going to see this mechanism again when we talk about insulin. And it's kind of similar, actually, to the mechanism for PTH as well. Not exactly the same. So you should probably know this. It's a hint. Hyperventilation. What will hyperventilation probably cause? Ooh, discuss. You're like 50-50. Now, what do you think? Does everybody agree with that? You all think that's the correct answer? Because I said so? You trusting fools. Think about it. What happens when you hyperventilate? What am I doing besides looking like a fool? Exhaling CO2. If I'm exhaling CO2, what's happening to the amount of CO2 in the blood plasma? It is decreasing. CO2 is a volatile acid. If I'm getting rid of acid, I'm causing an alkalosis. The answer should be B. Right, Blake? You really feel like it should be A? Nope. You're exhaling CO2 at a faster rate, so you're getting rid of it. So when you hyperventilate, how do you treat it? What do you do for somebody who's hyperventilating? Breathe into a bag. Give them a paper bag so that the CO2 that they're exhaling comes back in. So somebody hyperventilating, give them a paper bag and they breathe in the CO2 that they're breathing out so they don't go into alkalosis. Yes? No, hyperventilation can increase oxygen in the alveoli. If your breaths are shallower, then you're taking in less oxygen. Yep, so if your tidal volume is lower, so if you're going, you're mostly exhaling. We got it? Anybody still confused on this? Yes? Yeah, you're getting rid of CO2, so you're getting rid of acid. Oh. So you think I was correct in the beginning? Well, come on, help me out here. What does the question say? I don't know how many of you come into my office and look at your exams and say, damn it, I didn't read the question. So what's the question say? Is a cause of hyperventilation causes, right? Isn't that the same thing? No. Hyperventilation can cause? That would be hyperventilation is a result of, right? So that's why I put this up here. Some people get confused by the wording of a question. Right? So this basically says hyperventilation is a cause of. Hyperventilation causes. If I wanted to ask you what caused hyperventilation, I would say hyperventilation is a result of or results from. Respiratory alkalosis. Hyperventilation causes respiratory alkalosis. So hyperventilation causes, hyperventilation results in, hyperventilation is a cause of. So if you hyperventilate, the end result is this. Yes? Right, hyperventilation is a result of or is caused by, would be C. Yep. Right, so if you've got a metabolic acidosis, you've got too much acid and you've got to get rid of it. If it's metabolic, it's a bicarbonate issue. The kidneys are going to have to take care of it. Takes them a long time to do that. So the immediate compensation is going to be hyperventilation to get rid of volatile acid, CO2. So the fastest way to compensate for an acid-base disorder is changing respiration to get rid of or retain CO2. Right, because the kidneys take such a long time. The only problem that doesn't work very well is if the cause is in fact respiratory, then the respiratory system is not going to be able to compensate very well for it. Yes? So hyperventilation is a cause of, or causes, or results in. Yes. Yes. Yeah, they're expelling, hyperventilation by definition is expelling CO2 faster than normal. Hypoventilation is retaining CO2, not getting rid of it as fast as normal. So respiratory control, the primary drive is going to be CO2. Yes? Respiratory alkalosis, B. So changes in arterial PCO2 sensed by the central chemoreceptors are going to give you the greatest change in respiration. pH gives you a smaller change, so the cells are less sensitive to that. And PO2 is peripheral chemoreceptors. And the only way those peripheral chemoreceptors are going to respond is if you get a 40% reduction in PO2. So PO2 has to drop below 60. Normal lab values, carbon dioxide, 35 to 45 millimeters of mercury. So we say, in all the examples that we use, the normal PCO2 is 40. Oxygen, between 80 and 100. Normal arterial oxygen should be between 80 and 100. Peripheral chemoreceptor response will not kick in until that oxygen level drops below 60. pH, 7.35 to 7.45. Right, normal pH 7.4. Bicarbonate, 22 to 26 milliequivalents per liter. These are going to be important when we get into diagnosing acid-base disorders. So carbon dioxide response at different O2 levels. As O2 drops, you see this greater increase in the curve for stimulation of alveolar ventilation. So as O2 drops, you're going to see a greater sensitivity to changes in CO2. Under different conditions, this response curve is going to change. So normal, when you're sleeping, the response curve shifts a little to the right, so that means you can tolerate higher levels of CO2 than when you're awake. Things like narcotics and anesthesia are going to flatten out this response, such that large changes in CO2 are not going to stimulate a significant change in ventilation, which is how excess narcotics and anesthetics can cause respiratory distress. Of course, metabolic acidosis is going to give you the greater increase hyperventilation, right, to get rid of excess acid. So chemical control, primarily via CO2, central chemoreceptors. Secondarily, oxygen and hydrogen ion in the peripheral chemoreceptors. You also have non-chemical control, which we don't talk about too much in detail, so stretch reflexes, joint proprioceptors, etc. Higher brain centers for voluntary changes. So the effect of CO2 on ventilation. So a decrease in CO2 right, correlates with a decrease in ventilation. Increase in CO2 correlates to an increase in ventilation. So if CO2 increases, it stimulates the chemoreceptors to cause you to hyperventilate to get rid of that CO2. If CO2 is low, those chemoreceptors will respond at a lower rate. You'll get a lower action potential frequency going along the glossopharyngeal and vagus nerves. Oh, wait a minute, this is CO2, so this is central. Never mind. You get a lower action potential frequency coming from the central chemoreceptive area, and that'll cause an increase or a decrease in ventilation. So you've already got low CO2. You don't want to get rid of what you've already got. So hypocapnia is low CO2 and hypercapnia is higher than normal CO2. Problems with CO2, problems with acid-base balance develop when you have respiratory disorders. Most of the disorders you'll see either fall in the COPD category, chronic obstructive pulmonary disorders, and that's a large class of disorders which encompasses emphysema and a bunch of other different disorders. And then there are some other, and you'll deal with asthma a lot, right? So severe lung disease, you can develop hypoxemia, so low blood oxygen, and hypercapnia, high blood CO2, because you can't get rid of the CO2 and you can't get the oxygen. Right? There's a problem with that gas exchange process. If this is a chronic condition, then over time, the CO2 sensitive cells will adapt to that higher level of CO2, just like baroreceptors in people with chronic high blood pressure adapt to that higher blood pressure level. So it's kind of like resetting the thermostat when we talked about pyrogens in the brain, right? So if you have COPD or some other respiratory disorder where you have chronic high CO2, those receptors are then going to adapt to that higher CO2 and no longer respond normally. This is a problem because treating that with oxygen can inhibit the respiratory drive. So if your CO2 sensors are no longer responding, they're no longer driving respiration. Respiration is being driven by oxygen now because the CO2 sensors are no longer responding normally. So, under normal conditions, your respiratory drive is controlled by CO2 because your oxygen levels are 40% higher, right? They have to drop down to around 40 or below 60 before the peripheral oxygen sensors respond. Under conditions of chronically high CO2, so COPD, oxygen levels are going to drop, so not only are you having a problem getting rid of CO2, you're having a problem getting in oxygen because of the damage to the respiratory membrane or the problems with the lung tissue not being expansive or compliant. So at this point now, your CO2 in the blood is so high that the CO2 sensors no longer respond to it. They've gotten used to being at this high level. They're no longer responsive. So the only thing keeping you breathing at this point, and you will see patients who've been smoking for decades who are on oxygen therapy, right, who when they first come in might be in this situation. So if their respiratory, if the damage is so severe that their respiratory drive is due to oxygen, the last thing you want to do is give them oxygen. Giving them oxygen removes the respiratory drive, and they will stop breathing. Right? Their CO2 sensors are not responding. The only thing keeping them breathing is low PO2. If you raise their PO2 quickly, they're going to stop breathing. So the treatment for these folks is usually to try and lower the CO2 first, and then deal with, well, I jumped ahead. Okay. Which of the following blood gas values is most likely to cause hyperventilation? So this is now the flip of the question that I gave you before. Right? So in this case, the high PCO2, excess acid, causes hyperventilation for you to get rid of that CO2 and lower blood CO2 levels. So in this case, I flipped it. So in summary, take-home messages. CO2 is the primary regulator of respiration. Right? Central chemoreceptors are most sensitive to CO2, so that's where the primary response is going to be generated. There's a secondary response to oxygen in peripheral chemoreceptors, but there needs to be a large change in PO2 for that to occur. If the PCO2 is constant, low oxygen can be an important regulator of respiration if it falls below 60 millimeters of mercury. So a couple of questions to consider before Monday. Why do you think oxygen's effect on respiration is blunted? Why is it oxygen does not have as great an effect on respiration as CO2 does? Right? It takes a 40% change in oxygen to result in any change in respiration. But you can get a change in CO2 of about 10%, which could result in a change. And explain ventilatory drive during severe lung disease. So the example I gave you is COPD. Questions? All right. See you on Monday. Yeah, I'll put an explanation.
And what did she say?